used to, and that they just like open out.
Okay, happy Wednesday. We are actually, today is our 12th lecture out of 23. So after today, we're more than halfway. So whether you celebrate or that makes you sad, I don't know, either way. You do have your second exam on Monday. I'll put out an announcement about that, but that's going to cover chapter 5, metabolism, chapter 7, genetics, chapter 13, viruses, and we're going to finish up chapter 9 today with some of the chemical antimicrobials. So those 4 chapters, remember the homework is due on Monday at 1:30 as well. So work on that a little bit every night if you have a lot of that to do yet. Remember, if your overall homework score is below 80% at the end of semester, then you will only earn whatever 78% or whatever of those 60 homework points. So it's not gonna be a boost for your grade if you don't get up to that 80% where you get all 60 points. So it's to you want to work at it a little bit to make sure you get up to that 80% and don't procrastinate too badly there. So remember, a week from today, Wednesday, that's your mental health day. So we will not have class next Wednesday. And Tuesday, I have to teach for somebody else's lab. So my office hours won't start until about 10. So, if you want to come to your exam, at least wait till 10 o'clock there. It has taken the processing longer. I don't know if you pay attention, for me to get the lectures up, it's taken a little bit longer lately. I don't know why. There's some kind of processing that it hasn't shown up on my cloud till later. So I always try to get that up as soon as I can, but that's been out of my control. Okay. All right. So always don't be afraid to stop me if you have any questions. I know it's a little harder to ask questions when we're so spread out. But last time we did start thinking about antimicrobial methods. We spent a good bit of time on some applications of heat, radiation. We spent a little bit of time there. And remember, with our genetics, we talked about mutation kind of in terms of, oh, this hurts the bacterial cell. And this is what the cell does to try to, correct mutations. Now we're thinking about, yeah, we want to mutate the cell. We want to kill the cell. And what are some ways that we can do that? We're only talking about 4 different chemical antimicrobials. So the book talks about a lot more. I think the ones that seem to be in higher use, more applications. And we organize those, remember, in terms of kind of a low-level antimicrobial, medium, or high. So the soaps and the surfactants are considered low-level. Remember how they work, surfactant action. So they're going to work mostly on cell membranes and cell walls, disrupting those. And then we started talking about halogens, which are considered more mid-level. So on that chart of organisms, different microbes that are easy to kill and hard to kill, like we're getting a little bit higher up the ladder there. So medium level chemicals or antimicrobial agents are still not able to kill spores, right? Within a reasonable period of time. So, if we bleached, some surface for an hour, that would be sporicidal, but that's not considered a reasonable amount of time. So still considered medium level. The halogens like chlorine and iodine, remember those are strong oxidizing agents. So they're gonna denature proteins primarily, but causing other damage in the cell likely as well. All right, so chlorine, in the form of bleach is probably kind of the go-to thing, at least that I think about using when I'm trying to clean at home. It's a pretty strong disinfectant. It works really fast. So a lot of disinfectants, you need to, have some contact time. Bleach is one of the most fast acting. So usually only a few seconds. It's going to kill most of whatever's on that surface. It is considered a disinfectant. So remember the difference between a disinfectant and an antiseptic. An antiseptic is going to be only used on living surfaces. A disinfectant is designed to be used on non-living surfaces, so it can be, a little bit stronger. It has to be safe to be on our skin. Obviously, you can get your hand in some bleach, and it's not going to really cause you any problems. But you want to wash your hands in bleach all the time, or be constantly having your skin in bleach. That would be really irritating. So that's why it's a disinfectant. One drawback to using any type of chlorine is that it is susceptible to organic load. So remember, that's one of the things that we talked about. If you have, a dirty surface, or it's contaminated with some kind of bodily fluid or vomit or whatever, you have to clean it first and then disinfect it. A lot of cleaners don't penetrate through that organic load very well. And chlorine does have another drawback in that it's sensitive to both light and pH. So we mentioned that, if you're treating, adding chlorine to a swimming pool and the pH rises, the chlorine is going to dissociate and not work as well. It's not going to have as good killing power. That's why you buy bleach in these opaque bottles, because that light is going to hasten the decomposition of the chlorine, and it's not going to work very well. I always try to buy bleach in the smallest container that I can, because after about 9 months, it starts to dissociate and not work as well. So if you find, I use a little bleach, on wash rags or, laundry or whatever to whiten them. When I find that the bleach is not working to whiten the laundry, oh yeah, I've had this bleach a couple of years. Oh, okay, time to throw it out, time to get some new bleach. So if you've had it longer than a year or so, it's not gonna work as well as it should. So something to keep in mind. There's lots of different types of chlorine that's in use. And I'm not too worried that you kind of really know the difference between all those different types. Elemental chlorine is what's often used to treat water. So whether in swimming pools or sewage treatment plants, we add chlorine. Some type of chlorine is kind of that last stage of cleaning. If you see a lot of sewage treatment plants have these big pools. Well, you add the chlorine and then you have to let the water kind of sit there for a while for the chlorine to evaporate off so that you can release it back into the Grand River or whatever. So water treatment chloramine is the type of chlorine we see that's complexed with ammonia. So chlorine and ammonia. And that's a liquid. So you may have heard, oh, never mix bleach and ammonia. In fact, don't even have both those things in your house, because if you mix those two cleaners, it makes a toxic fume that has killed people. So it's really kind of hard to even find ammonia in grocery stores, probably for that reason. But if it's in the liquid form, chlorine and ammonia liquid, it's okay. It's when you mix the two separate things and it's the vapor that's toxic. When chlorine's mixed with ammonia, it's not quite as effective at killing, but it's longer lasting. So, you can add it to whatever water, or sometimes they dip dressings for wounds in chloramine, just, longer acting killing power. Still pretty effective. Hypochlorites, that's the fancy name for bleach. Okay, so probably, lots of different applications, certainly for bleach, cleaning all sorts of surfaces. In a norovirus outbreak, that's what you want to do. You want to use some type of bleach to clean, because those naked viruses need something pretty strong on there to get rid of them. You can also use chlorine in a gas form. So if you need to disinfect some kind of large space, that can be pretty effective. All right, so lots of applications with chlorine. Iodine, we tend to have, we think about using iodine on living surfaces. So more as an antiseptic because, you know, when you think of iodine, it's this brown liquid. Okay, it's going to stain pretty much any surface that it comes in contact with. So, you know, you wouldn't want to use iodine on your countertops or your floor at home because it's going to stain it. Yes, it stains our skin, but it wears off after a day or two. All right, so it does have the advantage of being a little bit less susceptible to organic deactivation, so penetrates a little bit better there. And we see using this in different forms as well. So sometimes it's dissolved in water, but it's a little bit more effective, a little better killing power when it's dissolved in alcohol. And we call that a tincture of iodine when it's dissolved in alcohol. So that's more commonly the way you see it. We can use this as wound treatments. Campers sometimes use iodine tablets to treat water to make sure that it's okay to drink. So, you know, instead of carrying a bunch of water when you're out backpacking, it makes sense to just have a container and some iodine tablets. It has the big drawback of here that it doesn't kill certain protozoa. And it's a protozoa called Cryptosporidium. So I'll just write that in. That Cryptosporidia is a kind of common contaminant of freshwater sources. There have been outbreaks of this in water treatment plants. There was one in Milwaukee in the 90s where like 40,000 people got sick with this really nasty diarrhea just from drinking their water out of the tap. Iodine, again, doesn't effectively kill that at the concentration that it's okay to still have us drink the water. All right, so we added really concentrated, it would, but not when it's okay for us to still kill the water, or sorry, drink the water. All right, so some disadvantages, you know, maybe a little irritation when you use this on the skin, some staining, minor allergies. Another form that we use iodine in is an iodophore. And that just means that we complex the iodine with some kind of a carrier molecule, and that makes it longer lasting and less irritating the skin. Okay, so that's probably more commonly if you've got, you know, you got to have a needle poke. They will usually clean it off with an iodophore. Again, it lasts a little bit longer, less chance that the patient's going to have a little bit of a rash as a result of that. Here, this picture, you see them washing somebody's hand in an iodophore, I believe, as just a way to clean off the hand before hand surgery. The trade name of iodophore is Betadine. Okay, so if you've ever seen that, Betadine, that's iodine when it's been complexed to a carrier molecule. So whether it's, you know, Betadine or tincture of iodine, they're all different forms of iodine. Another application of Betadine is in the dairy industry. So I found after watching some of my distant relatives at their dairy farm, I asked them about, they use something called teat dip. So basically they, at least my relative's farm, they bring like 5 or 6 cows into the milking shed at once. They hose them down a little bit because, right, they lay in manure. This picture is like the cleanest cow I have ever seen in my life. But they hose them down a little bit, and then they squirt this teat dip. You know, they go up and down the row, do it on all 4 of the teats, and then, you know, go back and wipe it off. So it has, you know, a minute or 2 of contact time to it, and then they wipe it off because they don't want the iodine dripping in the milk. So just another way that we have, you know, we talked about pasteurization last time, but another way that we have pretty hygienic methods of milking now. Still need to pasteurize, but it is fairly hygienic. Okay, another kind of mid-level antimicrobial chemical that's in lots of use, alcohols. So when we think about using alcohol as an antimicrobial, it's usually going to either be in the form of isopropanol, which is rubbing alcohol. That's actually the main ingredient in a very common cleaner, Pine Sol. That active ingredient in Pine Sol has changed over the years. It used to be phenol. That kind of fell out of favor, you know, too irritating, and it's changed over the years. But isopropanol and ethanol. Ethanol, same thing as ethyl alcohol. I was checking the ingredients on some hand sanitizer, and ethyl alcohol, same thing as ethanol. So the main thing we think about using alcohol for now is in hand sanitizers. Alcohol can be used as a disinfectant. It just kind of depends on the concentration. So if you look at your hand sanitizer, this one, it says the brand is Germex. How much alcohol? I believe it was 63% alcohol in there. So the hand sanitizers usually range from about 62% to about 16. I think Purell is more like 69 or 70%. All seems to be good, depending on what brand. How does that work? So it's got some kind of surfactant action, so it dissolves the membranes. When it can penetrate through those membranes, it's also going to denature proteins. So kind of in several different ways. I was reading, you know, how when you Google something and you kind of get down a wormhole, you know, there's all these things on the internet. Can you drink hand sanitizer? As you would expect, no, you shouldn't. I'll just throw that out there. It's alcohol, right? But it's actually a denatured alcohol. So you should never drink any alcohol that's denatured. I don't know exactly how they denature it, but in hand sanitizers, they add some additives in there, you know, to make it smell good usually. And it's those additives that can really make you sick as well. So short answer, no, you shouldn't drink it. I had a brand of hand sanitizer that smelled so bad, it made me nauseous. I had to throw it away. And I thought, why would they want to make it smell so bad? It may be as a deterrent so people don't think about drinking it. I don't know. But there you have it. When you're using alcohol, typically, they say that the 70 to 90% alcohol is most effective. So probably if you're thinking about more of as a disinfectant, we've got this other spray up here. It says it's hand sanitizer, but it says it's 80% alcohol in there. So that's a little higher percentage. Why do we always put some water in there? Some water is necessary for that surfactant action. So remember, surfactant kind of diminishes the attraction between molecules. So, you know, releases the bacteria from each other and is helping to dissolve membranes. Okay, so that's why we don't use 100% alcohol. So you need a little bit of water in there for that surfactant action. In addition, 100% alcohol evaporates really quickly. And you always want to have at least a little bit of contact time. Okay, so when we use it at the lower concentrations, it's not irritating to the skin. This brand says moisturizing. I don't know what they put in there. I doubt that it's really that moisturizing. It is going to dry your hands out a little bit. It's very cheap. Okay, so that's a good thing. If you use alcohol to really high concentration and it's a vapor, then that can be an inhalation hazard. It's especially kind of hard on your eyes as well. It can affect the nervous system. Okay, none of that at the concentrations that you get for hand sanitizer or, you know, spray on like the alcohol that we spray in labs. So you'd have to have a whole lot of it to be dangerous. All right, so we got to talk about hand washing while we're here. So what's better, using hand sanitizer or washing with soap and water? Well, let's define how we do both those things correctly. If you use hand sanitizer, you should use enough that your hands are going to be wet for 15 seconds. That seems like kind of a long time. Like, you know, you wash your hands and then you get ready, or you rinse, use your hand sanitizer, and then you go on to do something and your hands are all wet, and it's sort of annoying, but you need enough that it's going to be on there for a little contact time. And if you're hand washing, they say, you know, to wash hands correctly, you should, you know, scrub for about 20 seconds. I'm probably guilty of not doing it quite that long. But again, with those soaps, you need some, you know, mechanical scrubbing to get rid of the dirt that's going to take the bacteria and viruses with you. All right, so if you have the choice to use one or the other, it's always better actually to wash your hands. Now, if you don't have access to soap and water, hand sanitizer is a good plan B. But some drawbacks with hand sanitizer, make sure I say this right. It's not effective, or not as effective, when your hands are dirty. So again, those organics get in the way. There's no way to kind of rinse it off. I think back to a few years ago, I was on a mission project, you know, out in a third world country, and we were building these houses, and when we'd stop for lunch, oh, we'd be sure to, you know, use the hand sanitizer. Well, that was better than having clean water available to wash our hands, but there was no clean water available. So, you know, we thought we were really, you know, making sure our hands were clean. They, you know, cleaner than they were before, but probably not killing everything. Another drawback with hand sanitizer is that it doesn't reliably kill spendospores. That's not too surprising. That's probably not a problem. Or naked viruses. Okay, so remember the example of naked viruses that I keep going back to is that norovirus. Got a norovirus outbreak, hand sanitizer is not quite gonna cut it. Pretty good, but not, you know, that commercial about the mortgage commercial. It might be pretty good, but it's not, you can't be totally sure. So, you know, hand sanitizer, great to kill coronavirus, great to kill influenza, but not working on everything. So for those reasons, you know, it's probably best to wash your hands if you have the choice, if you have the chance. All right, our last antimicrobial chemical is considered a high-level disinfectant and antiseptic. And I've read that it's probably the closest thing that we have to kind of that perfect antimicrobial, and that's hydrogen peroxide. Okay. So why is it so effective? When hydrogen peroxide dissociates a little bit, when it decomposes, it forms hydroxyl radicals. And hopefully you recognize that as one of those toxic forms of oxygen. It's going to pull electrons off of other molecules. So it's an oxidizing agent. Okay, and it's going to denature proteins, break down membranes. Okay, lots of cellular damage there. One disadvantage to it is that it's photosensitive. So anytime you see something marketed in a bottle, you know, dark glass or dark plastic, that's probably a tip off that there's some photosensitivity there. It will go off much quicker when the sunlight hits it. So we try to keep it in a dark place. But It has a lot of advantages. It doesn't leave any residue, it doesn't damage surfaces, and it's really effective. So most commonly, hydrogen peroxide is used as an antiseptic at a pretty low concentration, somewhere like 3%. And this has fallen out of favor a little bit in terms of using it as an antiseptic. When I was a kid, you know, fell down and scraped my knee, we'd squirt hydrogen peroxide on there and it would bubble. You have to use enough so that it stops bubbling. Okay, so that bubbling is because our cells, as well as bacterial cells, are releasing catalase, which dissociates that hydrogen peroxide into water and oxygen. So those bubbles are oxygen. If you're in lab, you did that test last week. You have to use enough to overwhelm the catalase producing capacity of the bacteria. So use a good bit of it on there. And you shouldn't keep adding hydrogen peroxide to a wound day after day after day, because it will slow down the healing process. Okay, it's hard on our cells as well. So it retards the healing, and then the wound is more likely to scar. So it's good, use it once, and then maybe use something else, some other kind of antibiotic cream maybe, that's not gonna be slow down the healing process. Okay, we can use hydrogen peroxide at higher concentrations, say 30 to 35% as a liquid or even as a vapor. Again, that's going to be sporicidal. So that's why it's considered high level. It even will deactivate those spores in a fairly short amount of time, less than 10 minutes. Okay, so it can be used to sterilize, you know, delicate instruments. Sometimes when we put like cutting instruments in the autoclave to sterilize them, that dulls them. So we don't like to do that. So it's nice to have some chemical that we can soak that in to sterilize them. Okay, questions about anything? All right, so that will be the end of the material for our exam 2, right there. We'll draw the line there. Okay. All right, but I do want to press on. I want to talk a little bit public health here, a little bit, just because this is a interesting times that we're living in. I didn't post this graph, but I find it interesting because it compares what people died of in the year 1900 with what they die of now, or 10 years ago, 2010. It's a little bit different for 2020, but we'll talk about that. So if you look at what people died of in 1900, the first, these are all the top 10 things. The first 3 of them are all infectious disease. So pneumonia or influenza, respiratory stuff, tuberculosis, another respiratory infection, gastrointestinal infections. So things like typhoid, people, you know, drinking unsafe water that hadn't been treated, or some foodborne illness. Okay, then we've got heart disease and stroke, neuropathies, that's kidney disease. Oftentimes that can have a infectious reason behind that. Let's see, accidents, cancer, senility, and then one more infectious disease, diphtheria. Okay, the lifespan for the white male, that's what we have good data on, in 1900 was 47 years. So a lot of people, it's a little bit confusing. That doesn't mean that people lived till they were 47 and then they usually died. There was a normal lifespan, but so many babies died before they were, 1/3 of babies died before they were 5 years old. Okay, so balance that out with kind of a normal lifespan and it brings the average down to 47 years, I guess. Okay, in the year 2010, average lifespan for a white male was about 74 years. So we've made some big advances. If you look at infectious disease in terms of what people died of, you have to go all the way up here to pneumonia or influenza is, number 9 cause of death. And you'll see that people are dying less often in 2010 than what they did in 1900, okay? 2020, they're already saying that coronavirus is the number 3 cause of death in 2020. So the infectious disease is making a little comeback, but hopefully not for long. All right, and this other chart has the crude death rate. Again, anybody that's interested in going into public health, this is the sort of thing that you'll be interested in thinking about. So what are some of the things that have made our death rate go down over the last 100 years or so? Well, starting in 1900, we were starting to develop health departments in municipalities across the US. So we're getting a little bit more organized, a little bit more serious about thinking about health and how to help people be healthy. First continuous municipal use of chlorine in water in the United States. So around 1910, we were starting to treat our water. So this, this big downturn that we see between 1900 and 1940, mostly due to improved hygiene, right? We treat our water, we're not getting sick from what we drink or probably less often from what we eat. Of course, we had this huge spike, 1918, 1919, worldwide flu pandemic. I think the most recent number I read was about 700,000 people in the US were killed. So we're getting close to that number for coronavirus, like 50 million people worldwide, but about 700,000 in the US. So again, further decrease in the death rate from infectious disease. This last human-to-human transmission of plague, that's due to increased hygiene, really. Remember, plague is passed on fleas of rats. So, we're not leaving garbage everywhere in big cities anymore, cut down on vermin in general. And then here, 1940s, first use of penicillin. So, we talked about hygiene last chapter. We're going to talk about use of antibiotics now. It's another thing that's making us healthier. And then polio vaccine here, the late 1950s, the passage of the Vaccination Assistance Act, that was just a way to try to get more preschoolers vaccinated. Measles vaccine came out around then. So kind of those are the big 3 things, improvements in hygiene, antibiotics, and vaccines. And, we see a little uptick here. Not everybody's vaccinating their kids. I think that's getting a little bit better now. Antibiotic resistant microbes have kind of come on the scene a little bit more, but okay, sort of interesting trends to think about. Okay, and both of those graphs were in the United States. We'd probably have a lot of different numbers if we were considering worldwide. All right, so chapter 10, all about antibiotics. What are some of the things we're going to talk about? We got to talk about a little terminology, and I always like to talk about a little historical perspective, some famous people that have contributed to the use of antibiotics. Let's make sure we understand what kind of organisms make antibiotics. We're not getting them from horses or something like that. And then thinking about some considerations of, well, kind of in general, whether a drug gets okayed to use by the FDA, and how would doctors decide when to use what drug, all sorts of things like how do you administer it, and side effects, things like that. And then we're going to spend a good bit of time thinking about particular antibiotics. Have about 14 or 15 of them that we're going to look at specifically, and try to understand, their mechanism of action, how the antibiotic actually kills the organism, what does it target in those bacteria, and thinking about kind of advantages, drawbacks of each antibiotic, and any like thing that we especially use it for. Okay, and then we'll finish up our discussion on probably not next week, the week after, about antibiotic, a little bit on antibiotic resistance. Okay. So there's some terms that can be sort of confusing when we're talking about using drugs in general. All right, and a lot of these slides are kind of content heavy. I got a lot of writing on them already. It's mostly because I was afraid I would forget to write things down if I didn't have it up there already. So again, some just want to discern between some of these terms. If you're talking about using a chemotherapeutic drug, a lot of people associate chemotherapy with treating cancer, but really that's sort of a catch-all for any drug that's used to treat, any kind of disease, whether it's cancer or diabetes or whatever, the flu. An antimicrobial agent, then it's another kind of all-inclusive term, but this is for a drug that's used to treat some kind of infection. All right, so typically, that could be a fungal infection, it could be a viral infection, it could be a bacterial infection. Okay, so when we use the term antibiotic, that is a drug that's going to be used to target bacteria only, right? And at least in the traditional sense, we think about an antibiotic being produced by living organisms. Okay, we say that it's biosynthesized. Okay, and we're going to talk about what organisms do that, but as far as we know, it's either a bacteria or a fungi that's going to produce an antibiotic kind of as a byproduct of their metabolism. If we have an antibiotic that's been modified in the lab a little bit, we call that a semi-synthetic antibiotic. Okay, so that's one that we've taken the natural product from the bacteria or the fungi and then maybe change just a little side chain of it, something like that. That's pretty common. A lot of the drugs in use now are semi-synthetics. There are a few that are true, biosynthesized antibiotics. And then we have totally synthetic. They're still considered antibiotics, even though they've been totally synthesized in the lab and not by organisms. But again, they're going to target bacteria, so that's why we consider them antibiotics. It's hard to read my writing there. Target bacteria. All right, so who are some people that have been important in developing antibiotics? I kind of crammed a lot on this slide here, some pictures. One that we talked about already on the first day, I think his name was Paul Ehrlich. He's considered kind of the father of chemotherapy. So he's the first one, early 1900s that really thought of, okay, not just preventing a disease with a vaccine, but what are some drugs that we can use that actually might have a chance of healing the patient. And he got the idea from that because he worked in Robert Koch's lab. In his lab, they were developing staining techniques to see bacteria. And they had learned of different dyes that would only stain certain bacteria. So he thought, well, there must be drugs that would harm bacteria and not harm us. So he kind of coined that term, magic bullet. So he wanted a drug that would, again, harm the bacteria and not harm us. So a magic bullet would, again, harm your enemy and not your friend. So I'll put that term in there, magic bullet. Okay, and he came up with a drug that he called salvarsan. again, he was testing lots of different dyes to try to find a dye that was effective against the organism that causes syphilis. He found one that had arsenic in it that had really good activity. So the problem is it had arsenic in it. It was really harmful for us as well. But for a time, that was used to try to treat people with syphilis. Another major advance came in the 1930s with another German scientist, Gerard Domach. Make sure I spell this right. Okay, so Gerard, I think it's Domach is how you pronounce that correctly. Again, another German scientist. He was working with the German pharmaceutical company Bayer and systematically testing many different compounds to try to find one that was active against Streptococcus in mice. And he found one that he called Prontosil, and that gave way to a whole host of sulfa drugs. And these were really miracle drugs when they came out, but they were really only active against very specific infections, Streptococcus, maybe some staph, but they're credited with saving the life of Winston Churchill in the 1930s. He had a bad strep infection. Okay, so we still use sulfa drugs today. We'll talk about them a little bit more, how they work, but they were totally synthesized in the lab. And in 1929, Alexander Fleming, he was a scientist at Oxford University in England. So the story goes, he walked into his lab after a long vacation and there were some Petri dishes. Back then, Petri dishes were glass. So you had to, wash them out and use them over again. And before he left on vacation, he'd put some Petri dishes off to the side and didn't clean them off. When he came back, he noticed that on his plates with Staphylococcus growing on them, there was a mold that had this huge, zone around it that he noticed, oh, the staph are not growing close to that mold. That mold is secreting something that's keeping the staph from growing. So he did some tests for a while and, named this compound. It was a penicillin mold, Penicillium mold. And so he called it penicillin. But he didn't have any luck trying to purify whatever it was that was killing the bacteria. And he wrote it up in a paper, but he eventually just kind of moved on to something else. And then in about 10 years later, Howard Chayne, and that's him right there, and Ernst, sorry, I said that wrong, Howard Florey, that didn't look right, and Ernst Chayne. Howard Florey was, scanning the literature, trying to find some compound that might help with bacterial infections. They saw war on the horizon. They knew lots of people were going to die just of simple infections and not necessarily of gunshot wounds. And he came upon Alexander Fleming's paper and assembled a team around him. Ernst Chayne was an organic chemist to try to figure out how can we mass produce penicillin. So they worked pretty hard at it and finally found a way. Apparently you can only share the Nobel Prize between 3 people, and they chose Fleming, Florey, and Chayne, but there was lots of other people that made contributions there that don't get as much credit. All right, so where do antibiotics come from? We don't really talk about this in your book at all, but I wanted to make sure that everybody was on the same page here. All of the antibiotics that are in use are either secreted by some kind of bacteria or some kind of fungi. All right, so I just gave some examples here. They're mostly soil bacteria or fungi that live in the soil. Just some examples where we've gotten some medically useful antibiotics. Bacillus, it's a soil organism. It's a gram-positive rod. We use it in lab. Streptomyces is another genus of bacteria that's given us like almost half the antibiotics that we use now come from Streptomyces. And this is actually kind of a weird bacteria. This is a light micrograph here of Streptomyces. It grows in these long filaments, and it actually produces reproductive spores, which is not something that bacteria are supposed to do. That's supposed to be kind of a fungal thing, but they're prokaryotic cells. So it's kind of this weird cross between a bacteria and a fungi. We call it a bacteria because it's got, you know, bacterial ribosomes and no membrane-bound nucleus, but it's really kind of a weird organism, soil organism. And then fungi, of course, you've heard of Penicillium. Those form kind of greenish molds, usually with maybe some white edges. And then another one, Acremonium, that's this one on the right here. There's a whole class of antibiotics called cephalosporins that are secreted by the Acremonium genus. And that genus used to be called Cephalosporium, which made a whole lot more sense, but I don't know, they changed the name at some point there. Okay, so what do we need to think about when we are using antibiotics, or which ones to use? You know, are they safe? All these things we need to think about. The primary thing that we need to think about, and hopefully somebody else has thought about that before we have to make that choice, but we always have to ask the question, as antibiotics are being developed, are they selectively toxic? Okay, so that means, do they target bacteria without harming the host? Okay, so that typically means they're going to target some structure of bacteria that we as animals don't have. All right, so that's the primary consideration. If the drug is targeting, you know, a structure in bacteria that's absolutely the same in our cells, it's not going to be safe for us. So it's going to do, you know, just as much harm as good. There's other things we have to think about in terms of safety and side effects. Some people will have an allergic reaction to the drug. Okay, and we're going to talk a little bit more about that, but common allergic reactions would be hives or a rash. And it's only certain people. It's an inappropriate immune response that some people have to the drugs. Okay, you're going to have that with, you know, just about anything. Some drugs have toxicity issues. So even though, you know, a part of the microbe is targeted that we don't have, there might be some side effects from that. Kidney toxicity is probably one of the most common toxicity issues that we have to kind of be careful of. Also disruption of normal microbiota. So, and that plays into the spectrum of activity. So let me come back to that. Antibiotics can either be considered a narrow spectrum or a broad spectrum. So if you have an antibiotic that's narrow spectrum, that means it's only going to kill a couple of kinds of bacteria. It's going to be kind of specific. Like the penicillin only targets gram-positives. A broad spectrum antibiotic would kill probably gram-positives and gram-negatives, or at least a wider range of organisms. So you probably think, oh, well, that's great. If it kills everything, that's better. Problem with a broad spectrum of activity is that it's going to have more effect on the normal bacteria that live in our body, that we rely on, that we need to make us healthy. Okay, so when we cut down on that variation of our normal microbiome, then some things can overgrow that we don't want to overgrow. So we could get a fungal infection like thrush, right? A lot of us have a little yeast in our mouth just normally, but it's kind of kept in check by the bacteria in our mouth. If the bacteria for some reason are killed, then those yeast can overgrow. Okay, that can happen with, you know, vaginal yeast infections as well. So it may seem like broad spectrum is better, but it's actually better to have, as long as, if we know what we're trying to treat, it's really better to target just that thing rather than kind of killing everything else. Okay. Let me, before I get to the efficacy, I forgot to move on to the next slide when I was talking about selective toxicity. So here's some examples, sorry to back up for a minute, of selective toxicity. This is the graphic from your book. Okay, mechanisms of action of microbial drugs. So we're going to go through all of these and give some examples of each of them. We're going to spend a good bit of time on antibiotics that inhibit cell wall synthesis. So think about, that's great selective toxicity. Bacteria have cell walls, most of them do anyway, and animals like us don't. All right, don't tell me eukaryotes don't have cell walls. Remember, plants and fungi have cell walls. It's animals that don't have cell walls. So a lot of our antibiotics target cell walls. We're not going to cover all of these, but a good many. Another way that drugs can be selectively toxic is by inhibiting protein synthesis. Because remember, bacterial ribosomes are 70S, eukaryotic ones are 80S. So we can target those bacterial ribosomes, and we're going to cover some of those as well. We do have 70S ribosomes in our mitochondria, but usually we wouldn't use a high enough concentration or dose of drug to ever be able to penetrate into our mitochondria. So usually that's not a consideration. This one's a little bit harder, disruption of cytoplasmic membranes. Bacterial membranes are really similar to ours, so we have a little bit harder time getting selective toxicity there. We will talk about the polymyxins. There's one example there. Trying to inhibit some metabolic pathway that bacteria have that we don't. The sulfa drugs do that. We'll get to that. Your 6th edition book has these two actually separated out. This picture from the old edition has them together, but we could try to inhibit DNA synthesis, replication, or RNA synthesis, transcription. There are some differences, slight differences between the way bacteria do that and the way we do it. So we're going to talk about these two, quinolones and rifampin. And we're not going to worry about this last mechanism. Viral drugs might try to inhibit, antiviral drugs inhibit the virus's attachment to or some kind of recognition to host, but we're not going to worry about that. So there's some examples of selective toxicity. Okay, so to finish up this slide, we also have to think about are organisms susceptible? So there may be some intrinsic quality, like for example, the mycoplasmas, they are bacteria that don't have cell walls. So obviously they're not going to be susceptible to penicillin that targets cell walls. All right, so that's kind of a no-brainer example. We can test whether the antibiotic might be effective against the bacteria by doing a Kirby-Bauer test. Okay, so if you were in lab, you got to do that last week. If you're not in lab, I just want to go over briefly how we would do a Kirby-Bauer test. Okay, this is a disc diffusion test, we call that. Okay. And what you do, you streak the surface of your microbiological media with the organism that you're trying to test. Say it's E. coli or whatever. You make a lawn of bacteria, so you get real good confluent growth. And then you put your different discs that have particular antibiotics on that media. You incubate it and see what grows up. Okay, what you measure is this zone of inhibition. So one side of the zone to another, and you use that measurement to help us understand whether that organism would be susceptible or resistant or kind of somewhere in between to that antibiotic. All right, so if a patient with that infection, you know, would they be cured? Would giving that drug to that patient have some therapeutic index impact? Is what we're really trying to understand. Okay, so if it's growing right up to the disc here, then that organism's not being affected by that antibiotic, and we would say for sure it's resistant. But in general, there's some difference as to how big the zone is to whether a patient would be helped by that drug or not. Okay, so that's a Kirby-Bauer test, and you can do other tests to try to understand the exact concentration that might be necessary in order to kill the organism as well. We do have to keep in mind what concentration that we're using. Let's see. So remember, we want to try to use some concentration that's going to kill the pathogen, meaning the disease-causing microbe, but be safe to the host. Okay, so we have something called the therapeutic index. That's a number that helps us understand kind of how safe that drug is. So the way that that's calculated, you take the maximum dose of the drug that a patient can tolerate. So we call that the toxic dose. Or here they're playing basketball over there. I will talk to somebody about that because that can be distracting, especially during an exam. So I'll make sure that they're not playing basketball on Monday. So sorry about that. So therapeutic index, you try to determine, you take a dose that's toxic to kind of the average person. And of course, there's differences in people as to, you know, what they can tolerate and what they can't. So that toxic dose is at the point where there's side effects that are unacceptable. So for example, if you take a drug for strep throat that makes your hair fall out, that's really not an acceptable side effect to treat strep throat. If you're trying to treat cancer and it makes your hair fall out, that may be an acceptable side effect because, you know, it's saving the patient's life. So again, it's not something that kills you necessarily. It's when the side effects become not acceptable. And you compare that to the minimum dose that stops the pathogen growing. So we call that the therapeutic dose. The concentration that has to be in a patient's body in order to get rid of the infection. Okay, so when you compare those numbers, so say for instance, you throw in some numbers here to make this make sense. Say the maximum dose that a patient can tolerate is 100, whatever, micrograms or whatever, and it only takes 1 microgram to clear the infection, then that's going to be a therapeutic index of 100. That's generally considered a pretty safe drug. Okay, so the difference between what you know makes you ill and what you know makes it get better. So some drug, as that therapeutic index gets lower and lower and lower, then you have to be a lot more careful about administering the drug and making sure the dose is just right. Okay, so in general here, drugs with a higher therapeutic index are safer than ones with a lower therapeutic index. Okay, so once we get, you know, down to like, you know, it's hard to look up this for drugs, but once you get down below like 50 or so, then they start being really careful. Okay, and it's going to make a difference how you administer your drug based on the therapeutic index. Okay, so there's two things that determine how we administer. We can say based on the therapeutic index, and I'll explain that in a minute. Also based on, let me see how I can, susceptibility to stomach acid. Okay, so let me explain that. So there's your graph in your book here, your figure 1013 in your book here talks about 3 different methods of delivering drugs, all right, or administering them, all right? So you could administer them orally. That just means you take it in a pill or liquid form. You can see that the concentration in the blood doesn't really get up very high when you administer it that way, but it's certainly kind of the cheapest and easiest way to administer drugs. We could administer it intramuscularly, so by a shot. You get the concentration up a little pretty high right to begin with, but it often dissipates really pretty quickly. And you can also administer by IV. You can see with that way, you can really dial it into whatever concentration you want. The other method is just topically, right? So there's some drugs we just put on our skin. All right, so if you're thinking about therapeutic index, if you've got a drug where you have to dose it out really carefully to make sure you don't get unwanted side effects, you're gonna wanna administer it probably by IV, okay? You don't wanna trust a patient to take a pill at the right time or not to take too many because it could, deliver a toxic dose. And maybe you could deliver it by shot, but it might not be as hard to, pin down what concentration that's going to give. All right, the other thing that you have to consider is the susceptibility to stomach acid. Some drugs, when they pass through the stomach, get degraded by stomach acid. So they're basically, worthless when they are going to be absorbed out into the tissues. So if it's susceptible to stomach acid, then you can't administer it orally. You have to give it either by IV or by shot, okay? Some drugs are going to have resistance, either intrinsic resistance or acquired. All right, intrinsic resistance, that just means they don't have the structure that's targeted. So I don't have a cell wall, so they're just intrinsically resistant. Acquired resistance, typically, we talked about this a little bit in our horizontal gene transfer. They can acquire some DNA, maybe a plasmid from another cell that gives them the instructions maybe to make some kind of enzyme that now allows them to deactivate the drug, for instance. So this is what we really worry about, the ability of big populations of drugs to now suddenly become resistant and our drug's not working anymore. And ideally, if we're producing antibiotics, we want them inexpensive. If we're gonna design the ideal drug, right, you might as well make it cheap to make. Okay. So all those are things that you're going to want to think about. All right. There we go. All right. So starting our kind of mini pharmacology course here, the first large group of antibiotics I want to talk about are ones that inhibit cell wall synthesis. Okay, and there's a huge group of antibiotics that do this. They're called the beta-lactam antibiotics or beta-lactams. Okay, so in general, I don't want you to memorize structures of drugs. Going to pharmacy school, maybe you want to do that. But there are some things in structures that you're going to want to recognize. And this whole class of drugs that are called beta-lactams, they're called that because they all have this beta-lactam ring. All right, so that's highlighted in green, and all these, you know, penicillins, carbapenems, cephalosporins, these are all beta-lactams because their activity rests in that ring. If you disrupt that ring, then they're not going to be useful anymore. Okay, so again, there's just some examples there. And if let's think a little bit why that structure is important. Okay, this is your figure from your book here that talks about how the beta-lactams inhibit peptidoglycan formation. So back in chapter 3, we spent a little time thinking about how the cell walls grow and how that has to be kind of a careful process. Certain bonds cut right along with other bonds being formed. It has to come about really carefully. So in this top part of the diagram here, this is how it's supposed to happen. So here's the bacterial cell wall. Remember, it's made with alternating N-acetylglucosamine and N-acetylmuramic acid, those sugars, alternating sugars. And then you have these cross bridges, these amino acid cross bridges that kind of stabilize the whole structure. Okay, so when the cell grows, some of these bonds have to be cut. The glycosidic linkages are cut by autolysins. And then other enzymes called transpeptidases. Okay. Those are the enzymes that reseal these amino acid cross bridges. Okay, so new sugar subunits are inserted after some of those bonds are cut, and then those cross bridges are again sealed up again with transpeptidases. If there are all these little green dots here, that is some beta-lactam antibiotic that's in the medium that the bacteria are in. If that antibiotic is present, then, you know, after the cutting happens and the new subunits get put in, you can see some of those amino acid cross bridges don't get reformed. That's because the antibiotic is binding to those transpeptidases. And so the cell wall loses its integrity, and it's really likely to burst due to osmotic pressure. So the cell is typically going to be in a hypotonic medium, lower salts in the medium, higher water. So water is going to rush into the cell. An intact cell wall would normally be strong enough to keep anything from bursting there. But if the cell wall is not intact, then you're going to have pretty commonly lysis of that bacterial cell. Okay, so going in a little more detail, that your book doesn't talk about this, but if you look at structures here, here's the structure of penicillin on the right, and compare that to the structure of D-ala-D-ala. So 2 amino acids, alanine. Amino acids are isomeric, so they have kind of a right-handed and a left-handed form, most of them. This D form is not typically found in proteins. That's kind of a form of amino acids that we see in bacterial cell walls. I think that's one of the only places. And those amino acid cross bridges can be of different amino acids, but most commonly it usually has these 2 alanines. And if you superimpose penicillin with the alanines, look at them superimposed. They fit exactly on top of each other. Okay, so actually penicillin acts as kind of a competitive inhibitor. I'm going to say like a competitive inhibitor, because we don't consider that because it's permanent. Typically, remember, a competitive inhibitor fits into the active site of an enzyme and blocks the substrate from binding there. That's essentially what happens here, but we don't consider that because it doesn't come out again. It's a permanent thing. Okay, so penicillin mimics the structure of these two amino acids. So penicillin binds to the active site of transpeptidase instead of those amino acids. Okay, so those cross bridges don't form. Keep in mind, this penicillin is only going to be effective in growing cells. So when a cell culture is in that log phase, that's when it's most susceptible to a drug like penicillin and other antibiotics as well that need a growing cell. Okay, and again, it's not really the penicillin that kills it directly. It's due to the influx of water that's the lysis that's going to kill the cell. Okay. All right, so of course we have to talk a little bit about penicillin. That was the first antibiotic that was discovered, again, Florey and Chain. And they had different versions of penicillin. Penicillin G, that was the 7th version. So it's the 7th letter of the alphabet, I think. The one that seemed to have the most activity against gram-positive organisms. Again, gram-positives, more peptidoglycan, makes sense that it would work against gram-positives. It has one advantage to it. It had a really low cost. Florey and Chain argued about whether to patent it, and in the end, they decided not to. So it made it available to, lots of people right away at a very low cost. So that's really to their credit that they didn't do that. That would never happen today. It reaches effective concentration into the tissues fairly quickly. It penetrates well there, and there's not many side effects. So those are all advantages. Disadvantage, this is really not used anymore at all because there's so much resistance to that. Bacteria, many of them have gained the ability to make some beta lactamases. So we're going to look at more closely the way that enzyme works in a later slide. When this first came out, it required injection because it was active acid label, meaning stomach acid basically broke it down. So broke the drug down. Okay, so we couldn't just swallow it because the stomach acid would destroy the activity of the drug. And it was absorbed pretty poorly through the small intestine anyway. So even if it did pass through the stomach, you would have to take a whole ton of it because only a very small percentage of it would get absorbed through the small intestine. Okay, but this was a miracle drug when it came out. There's no doubt about it. It saved millions of lives in World War II. And it's the later versions of this, there was penicillin V is the form that's still in some use today. I know when my son had his wisdom teeth out, they gave him penicillin as a prophylactic just to make sure he didn't get an infection. And I was kind of surprised. I thought we didn't just throw that around prophylactically anymore, but anyway, it worked. And so penicillin V has just a really small change to the structure. It's just like one molecule here or there, and that enables it to be able to swallow. Okay, so we can take it as a drug or we can take it as a liquid, and it doesn't have to be injected. So you may have had penicillin that didn't have to be injected. Okay, we'll pick up there in a week, hopefully get through the rest of our antibiotics, maybe even talk about resistance a little bit, but I think I've given you enough to think about. See you here on Monday for the exam. Have a good weekend.